So we are going to do the third part of chemical level PowerPoint. So we are going to talk about proteins at this instant. Let's take a look at some of the functions associated with proteins. Proteins are one of the most important category of organic macromolecules in our body. One of the most important role they have in our body is associated with their ability to act as enzymes and basically carry all of the chemical reactions in our body. We have millions upon billions of chemical reactions happening in our body. And as a result, we are desperately in need of functioning enzymes, which are also, again, as I mentioned, categorized as proteins. Proteins in general also have roles in creating your nail, hair, tendons, and skin. So majority of the structural functionality are associated with proteins. They transport majority of the molecules in your body, such as hemoglobin transporting oxygen, and the lipoproteins that I mentioned on the second PowerPoint, carrying out cholesterol or triglyceride. It also acts as the defense of your body. They act as antibodies to protect your body against foreign material, and they also act as hormones to basically um, carry out and control the activities of the body. Such hormones include insulin that control your blood glucose and growth hormone. They're also an essential component for your motion and movement, as your skeletal muscle is made up of proteins. Now, if I look at proteins, proteins are identified as a macromolecule, and they are made up of individual molecules referred to as amino acids. There are a total of 20 different amino acids within our body. 20 different amino acids. All of these amino acids can be combined in a different orders and with different length to produce hundreds upon hundreds of different type of proteins. So amino acids are molecules that through their different order and numbers can create a variety of proteins, and each of those proteins will carry a different function in your body, as I mentioned in the previous slide. If I'm taking a look at your amino acid structure, so looking at individual amino acid and comparing them together, these 20 amino acids, what you will observe is a central carbon, and on one side you have an amine group, which is NH2. On the other hand, you have a carboxyl group, which is COOH, and on the third part you have a hydrogen group. These three that are highlighted right now, they are all the same or remain the same among all the 20 different amino acids. So amine group, hydrogen, and carboxyl group does not change among the 20 amino acids. What does change is the R group you see right down here in red. So what differentiates one amino acid from another? It's not the presence of the amine or carboxyl group or hydrogen, rather the differences in the R side chain or the R group. Here I'm providing you with some example. We have valine right here, this is the R group, which is significantly different from glutamic acid, which is significantly different from lysine. What you see in blue in these three pictures are the R sites, and you notice that the remaining part of the structure are identical between these three amino acids. Now, when you use these 20 different amino acids and you order them in different numbers and different lengths, what you get is a protein. Now, how does a protein function is determined? We listed a lot of functions for proteins, but how do we determine the function of a protein? What determines the function of a protein is its shape. What determines the function of a protein is the shape of that protein. So how do I get a shape of the protein right? So to create a protein that is a functional protein, we undergo 3 up to 4 levels of organization. So when you originally connect the amino acids together to create a protein chain, that is referred to as a primary structure. Primary structure, as you can see in this picture, is just a long chain of amino acids. Each of the pink or purple circles represent an individual amino acid connected together via means of a covalent bond, which we refer to it as a peptide bond in the case of proteins. Now, if I start to fold this primary structure using a specific affinities or chemical bonds called hydrogen bonds, I can create 2 primary structures, or should I say, you can create 2 structures within the chains of amino acids. These 2 structures are alpha helix and beta sheets. So take a look at this picture. This is your alpha helix. You can see basically the helical structure. And this is the formation of the beta sheets within the structure, both created using hydrogen bonds. Now, if I take the secondary structure and I start to fold it even further and basically hide the parts that do not like to interact with water, water-hating parts, or hydrophobic, place those sections in the middle, and the parts that like to interact with water, put them on the outer aspect of the body as an hydrophilic region, I can create a structure called tertiary structure. So tertiary structure is the farther folding of your protein to place the regions that do not like to interact with water in the middle, and the regions that like to interact with water on the outer edges of the protein. I'm giving you a reminder that your body is 60% water. So anything that doesn't like to water is going to try to hide. And therefore we give it the name hydrophobic, as in they are afraid of water, like phobia. Hydrophilic implies water loving. They like to interact with water. Now, in many cases, proteins are done and are ready to be used or carry out their function at the tertiary level. However, there are sometimes that there are proteins that are relatively large, made up of thousands upon thousands of amino acids, and it's very difficult to form one single structure that represents a functional protein. So what happens is that multiple tertiary structures will come together to form a complex protein. So you will basically take multiple sequences of amino acid, undergo secondary and tertiary structures for each of them, and then you bring all of them together to form one functional complex protein. So here is the folding for the tertiary structure. So this is one tertiary structure. But notice that here in the quaternary structures, I'm bringing 1, 2, 3, 4 tertiary structures together to form one functional protein. So 4 of these structures have come together to make one functional protein. The best example that it represents this concept of the quaternary structure is the protein called hemoglobin that is made up of 4 globin subunits. Globin subunits are tertiary structures that are combined together to form one functional hemoglobin. Please take a look at the video I have down here to help you understand the concept a little bit in more detail. Now, proteins carry all the functions or majority of the functions of our body. They have a structural role and they all act as, not all of your enzymes are proteins. Now, because of these functionality and the tertiary, secondary structures, these proteins are very sensitive to the changes in their environment. So they would like to work in a constant environment. Going back to the whole concept of homeostasis and maintaining internal environment, one of the reasons we need to keep and maintain our internal environment is because to make sure that proteins are functioning properly. So in this case, proteins are functioning under a specific pH and temperature. If temperature and pH goes above or below the acceptable range or optimal environment, then protein shape is lost. And if you guys remember, shape is directly related to function. So with the loss of shape, you lose the function of the protein as well. Best example can be described as egg white. When you basically warm up your pan and you throw your egg in there, you see that your egg white starts with a very kind of clear material, and as soon as you basically expose it to heat, it changes to white and it never goes back even if your egg is cooled down. This is one of the reasons doctors are afraid of conditions where the person has high fever for a long period of time, because again, the chances of these proteins being denatured will be higher. Now, as we're talking about proteins, the concept of enzyme comes into play. So let's talk about enzyme. Whenever we carry out a chemical reaction, you carry out a chemical reaction by starting with reactants, basically your startup material. You need to input energy, which we describe right here as activation energy, for the chemical reaction to happen and create a final product. Now, this chemical reaction is required to destabilize what the bonds that are currently existing, and that energy is referred to again as activation energy. This is a significant amount of energy. Activation energy is a significant amount of energy that's required. And if you want to let this happen, basically you will not be able to carry the chemical reaction on the speed you require to basically survive. Because you are dependent of chemical reactions, right? So you need to carry these on a very specific speed, relatively high. But if these chemical reactions want to happen on their own speed, it takes a long time. Therefore, the chance of survival for that organism basically is non-existent. So what is the solution? The solution is use a catalyst. A more specific catalyst is organic catalyst that is going to speed up the chemical reaction by lowering the activation energy and again speeding up the chemical reaction. Here is the original scenario I described to you when there is no enzyme present. Here, when the enzyme is present, your activation energy has been lowered significantly, which means the speed of the chemical reaction will increase, and therefore we can carry these reactions at a much faster speed and therefore able to survive. Another name, as I mentioned earlier, enzymes are referred to as biological catalysts, something that speeds up the chemical reaction up to millions of folds. Here is if you take a look at an enzyme and its substrate. In this case, the enzyme has this region right here, basically these carvings. This region is referred to as the active site. This is the region where your substrate is going to bind to. In this case, maltose is your substrate, comes in, binds to the active site, where the chemical reaction gets broken between the molecules of maltose to produce two separate glucose. Again, these enzymes are specific. Their function is determined by its shape. So this enzyme will not be able to do anything else other than breaking down maltose to glucose. Enzymes can lose their function. They can denature if the environment includes high pH or or significant change in temperature. Sorry, I should say fluctuation in pH and fluctuation in temperature or exposure to radiation can cause damages to these proteins, causes them to denature, lose their shape, and as a result lose their function. Here is another example where you actually have two substrates. They bind to the active site. You create the chemical reaction to form a covalent bond, release water, the dehydration synthesis reaction, and you create your covalent bond right here. I really like this picture because it kind of highlights the idea of an enzyme and its functionality. So it says, it has nothing to do with you, Bessie. It's just that I'm lactose intolerant. If you don't know what lactose is a sugar found in dairy product. You need an enzyme called lactase to break down lactose to its simple sugars. So if your body is lacking lactase, the enzyme, you will not be able to basically digest lactose, and as a result, you get the symptoms of diarrhea, bloating, flatulence when you consume dairy products. Last group, I'm going to actually continue with this and finish it up. Last group of your organic molecules is a nucleic acid. Acids are basically DNA and RNA carrying your genetic information. Nucleic acids are made up of 4 individual molecules that can be combined together to form complex nucleic acid macromolecules. These individual molecules are referred to as nucleotide subunits and are made up of 3 components. Phosphate, a sugar group, it's a 5 carbon sugar group, which we call it a pentose sugar, and a nitrogenous base you see in a green box. The nitrogenous base option is adenine, guanine, thymine, cytosine, and uracil. We usually represent these by a single letter, A, G, C, T, and U. Now, when we talk about genetic material, something that I need you guys to notice again, it's not on your PowerPoint, but you do need to know that it is the 2 groups DNA and RNA. If we're talking about DNA sugar is referred to as deoxyribose, as in lacking an oxygen. In an RNA molecule, you have ribose as your sugar. Another main difference between your DNA and RNA is the bases that are used to make your nucleic acid. In the case of DNA, you are limited to A, G, T, and C. But in the case of RNA, your bases are A, U, G, and C. So the difference is the presence or absence of T. Last thing is that DNA is the original copy of the genetic information that is protected within your nucleus. However, your RNA is your RNA is a copy of your DNA and can come out of your nucleus and basically used to make copies of your protein. Please keep that in mind, the differences between DNA and RNA. In this case, the phosphate groups are identical for both. Lastly, I want to mention the concept of ATP. ATP is it stands for adenosine triphosphate. It contains an adenine, which is your base. It contains a 5-carbon sugar, and it contains 3 phosphate group instead of 1. The energy that is released and is used by your body through the process of through any process, if it requires energy, the energy is actually stored within the chemical bonds between the phosphate groups. So if I have 3 phosphate groups, I refer to this as ATP, as a T stands for, sorry, I'm going to write this down, tri, which is equal to 3. On the other hand, if I break down one of these, what I do is I get an inorganic phosphate. You have 2 phosphate now attached, so I can call this ADP, and that release of phosphate release about 34 kilojoules of energy. I can go a step further and break down the second phosphate, so I would release more energy, and now I will get AMP, which is adenosine monophosphate, as I only have 1 more phosphate group attached to this. For this part, please note the differences between DNA and RNA, and also understand what is ATP, and that's about it.